All right. Well, welcome everyone to what's going to be a toasty day in Davis. Not quite 100 today, but probably over 90. So what I'm going to do is basically one long argument here, and stop me anywhere along the way. The main point here is to try and convince everybody, or probably everybody's already convinced, so to get everybody on board with the concept of trying to do a broad phylogenetic survey of eukaryotic genomes, in particular microbial eukaryotes, because they're the ones getting ignored. We've tried to do this before, as I will tell you about, and the poor eukaryotes were nixed. I think the time is right to do this again, try this again, given costs of sequencing now. So the problem is pretty simple. Most people here probably understand this, but I'm just going to go through this. So I consulted last night the trusty Google and did a search for eukaryotic genome sequencing projects to try and find a list of eukaryotic genome sequencing projects. Of course, it's impossible to keep up with genome sequencing projects because there are so many, but there are a couple of lists out there. Wikipedia actually does a remarkably good job at keeping track of genome sequencing projects, although they sort of split them up into different categories and force you to delve into different pages, so that wasn't quite what I wanted here. So I went to GOLD, the Genomes Online database, which is a really amazing resource for tracking past, current, and even future genome sequencing projects. It's largely focused on bacteria and archaea, but they do have a pretty good compilation of what's going on in the eukaryotic world. So I went to this database. It's maintained by Nikos Kirpides at JGI. It really is a spectacular resource. I went to this database, and they have taxonomy. You can go to, there's a phylogenetic status of genome distribution, and they have them sorted by phyla. So here's the Archaea with the relative number of genome projects by Archaea phyla and bacteria phyla and eukaryotic phyla. Since I was concerned here with eukaryotes, I zoomed in on the eukaryotic group. And here's the list that they have. There are 704 of them that are unclassified. I didn't delve into those. I'm not quite sure whether that's because there wasn't obvious taxonomic information or because the NCBI taxonomy for those might be messed up. I don't know the details. But I took the rest of the genomes that are in Genomes Online. It seems like, it's not a perfect list, but it seems like a reasonable sampling of genomes. Here are the phyla distribution. So then I wanted to map the phyla. I don't know any eukaryotes. I didn't know what any of these things were. I wanted to try and map them onto a tree because I use trees for everything. Names are awkward. So I went in the literature to look for new, relatively recent papers on eukaryotic phylogenetic diversity. I found a couple. I looked at this one for a little bit, but I eventually settled on one with pretty images from Laura Parfait. She's here, right? Somewhere. Oh, nice paper, by the way. This figure was a little too complex for me. Fortunately, at the end of her paper, she has a really nice sort of consensus phylogeny with a little less names on there, or fewer names on there. And so I took her tree, stretched the branches out a little bit, and then mapped these phyla onto that tree. So many of the phyla are from fungi. 50% of the genomes in Genomes Online are from fungi. So I made a branch that corresponded to total 50 units of branch width for the whole tree. This is 25 units basically there. The next biggest sampled group is the animals, which represent about 25% of all the eukaryotic genomes. So I made a branch for animals that corresponded to that width. Next are plants and algae, listed in her tree as green algae including plants. That's 20%. Apicomplexans represent 5%, and then everything else is in the remainder, basically 0 or 1%. So not such a good sampling of the eukaryotic tree here in terms of genome sequencing projects. Again, it's sort of obvious. Most of the people out here, I assume, know this, but it's pretty annoying if you want to have good representation of genomes for a variety of purposes. So what is the solution to this? The solution is to fill in the tree, right? The solution is to use the tree to guide us and to go through the tree and select a better representation of organisms and sequence their genomes, if you want a phylogenetic sampling of genomes. So what I'm going to do now is tell you about my past experience with trying to get phylogenetic sampling of genomes, and both what we did for this, for other organisms, and why we did it, and then tests for whether or not it was actually beneficial for a variety of purposes to have phylogenetic sampling of genomes. I mean, I'm an evolutionary biologist, so I can get up and say I want phylogenetic sampling of genomes, but with the new lovely congressional efforts to have Congress people review NSF grants, that's going to be challenging, right, to just say I want phylogeny. And of course, that's challenging anyway. We need to show why phylogenetic sampling is a useful thing. So that's what I'm going to tell you about. And this, sort of my effort in this started many, many years ago. I was at Tiger, the Institute for Genomic Research, for 8 years, sequencing genomes. And Tiger actually, in their sort of after they sequenced the first couple of genomes, Haemophilus influenza and Mycoplasma genitalium, they got a large Department of Energy grant before I got there to sequence genomes that were largely selected to cover physiological and functional diversity, but there was a big component of that project that was trying to capture a little bit of phylogenetic diversity in the genomes that they were sequencing. And that was basically the last large-scale effort to cover phylogenetic diversity. After that, there were many large-scale efforts, the Gordon and Betty Moore Foundation for doing microbes, the NHGRI Human Gut Microbiome Project, the Fungal Genome Initiative, and so on. Lots and lots of projects that were largely focused on either pathogens or some type of functional diversity, but phylogenetic diversity was basically getting ignored. And I was, this was in about 2001 or 2002, and there were lots of people at many meetings and in papers who lamented this lack of a phylogenetic approach. Carl Woese even wrote about this, and Norm Pace, and many other people. And so we decided to look at the phylogenetic sampling of genomes and try and correct this. And right when we were doing this, fortunately, Phil Hugenholz wrote this really great paper in Genome Biology about the phylogenetic sampling bias of genome sequencing projects. So basically, at the time, he focused on bacteria and Archaea, and I'm just going to show you what basically was there for bacteria. At the time, there were about 40 phyla of bacteria that were recognized, 40 major lineages of bacteria. And almost all of the genome sequences came from 3 of those phyla, the Proteobacteria, the Firmicutes, and the Actinobacteria. And there was a scattering of genomes from the rest of the tree, but very sparsely sampled. And in most cases, phyla were represented, if at all, by 1 or 2 genomes. And the Proteobacteria at the time had about 100 genomes. So there was an enormous bias in the sampling of bacteria in the tree of life. This made me very sad. The same thing was true for other taxonomic groups. And I'm just showing you what the case is for bacteria, but the same was true for Archaea, and the same was true for eukaryotes at the time. So we decided that it would be really good to correct this bias. Me and Frank Robb and Naomi Ward and Karen Nelson put in an NSF Tree of Life grant that we thought there was no chance of getting funded to sequence genomes of the phyla of bacteria, representatives of the phyla of bacteria for which there were no genome sequences and for which there were cultured species that we could readily grow and get the DNA from. And amazingly, we got funded to do this. And so what we targeted were these 8 phyla of bacteria, and we sequenced genomes from a representative of each of these 8 phyla. In case you don't know, Tiger kind of exploded. Craig Venter moved back to the Tiger enterprise after he got kicked out of Celera, and many of the tiger people did not get along with Craig very well. I actually did, but I was in the very, very, very small minority. And eventually, Tiger kind of exploded. The Venter Institute took back over. Most of the Tiger faculty left and moved to various places around the country. I moved to UC Davis at this time, unrelated to Craig taking over. I just wanted to be in Northern California, basically. And our project kind of died halfway through because I left, everybody else was leaving Tiger, the whole system sort of fell apart. But we did sequence these genomes and do a few interesting, what I think, interesting studies based upon these genome sequences. And I'm not going to tell you about that, but I think we showed that having phylogenetic sampling of genomes was a pretty good thing. However, really, at the end of the project, we didn't have that big of an impact. And I think the reason we didn't have that big of an impact, if you think about questions about evolution, questions about the phyla of bacteria, questions about diversity, we were sampling a single genome from these phyla. Each of these phyla likely represented, 1 billion years of evolution, and we had a single genome from them. We couldn't, for example, ask questions about monophyly. We couldn't even test whether or not the phyla were real because we only had one representative from each of them. So, for example, at the time, this was a list, and I'm not even showing the same thing, of the major sublineages of Actinobacteria. I mean, we would have to sample each of the subgroups within these major groups to really have a handle on genomic diversity. And the same trend was true after we finished this project about Archaea and Eukaryotes, and although nobody has really tackled this, the same trend is true about viruses. And so the solution to this problem is to not just make some arbitrary cutoff and say, let's just get a representative of each of the phyla that we can get. The solution is to really use the tree as guidance and fill in, start walking our way through the tree and finding branches for which there are no genomes and filling them in and getting genome sequences from those branches. And we weren't the only people who sort of came to this conclusion. The NHGRI, for example, made this argument for vertebrate genomes in comparison to the human genome. Many of the plant genome sequencing efforts, in particular via NSF and JGI, said we need phylogenetic sampling of plants. The fungal genome initiative that was coordinated largely at the Broad Institute and a variety of other places said we need phylogenetic sampling within the fungi. This was not a unique argument, but microbes were kind of treated differently when we proposed these things. And one of the main reasons, which I'll get back to in a little bit, for why microbes were kind of put on a lower plane was the argument that many people said that lateral gene transfer wipes out all phylogenetic signal in microbes. And therefore, using a tree of organisms to guide your sequencing is completely meaningless because, in fact, there is no tree at all. We got this response many, many, many times. That is, there is no phylogenetic consistency. If you try and walk your way through a tree, it's completely useless. And so, I never bought that argument because I believe that, and there's evidence that there is some consistency within lineages. That is, they don't swap genes across all of diversity at an infinite rate. There is some vertical evolution consistency that people had been seeing within lineages, but we ran into this argument all the time. So nevertheless, when I moved here to UC Davis, I had an adjunct appointment at the DOE Joint Genome Institute, and I was actually at a meeting, a very boring bureaucratic meeting. I don't even remember what it was about, sitting next to Eddie Rubin, who's the director of the Joint Genome Institute, and he was actually also bored at this meeting. And he had an email that he had gotten from, someone associated, I think it may have been someone from the Burgess Manual Trust. I'm drawing a blank on exactly whether it was George Garrity or someone else, that said, basically we need a project to sequence all of the type strains of bacteria and archaea. We want to fill in the Burgess Manual and sequence every single type strain. There are only 6,000 or so of them. That seems like a reasonable thing to do. And Eddie gave me this email that was printed out with a note that said, what do you think? And I read it and wrote back and said, sounds great. Let's start with the phylogenetically diverse ones first. we should go use phylogeny. And literally, Eddie thought that this was a decent idea. I mean, he didn't give me money to do this immediately, but he said, that seems like a good idea. Why don't you talk to Nikos Kirpides and Phil Hugenholz at the JGI who do phylogeny stuff and see what they think. And of course, Nikos and Phil had been thinking about this exact same thing. It was kind of an obvious stuff, and we had been using Phil Hugenholz's paper from years before. And when I went to talk to them, amazingly, Hans-Peter Klenk from the DSMZ German culture collection was there in the hallway visiting the JGI. And we were discussing this project with him too, and he said it sounded like a good idea. And eventually, Eddie sort of made us go around to the community and garner support for this concept. We met with the Bergey's Manual people and said, we like your idea, but it's probably we think phylogeny should be the driving force, not sequencing everything. And eventually, we came to an agreement that we should fill in the tree. Fill in the tree of bacteria, fill in the tree of Archaea, and fill in the tree of eukaryotes. There were enough reasons to think that phylogeny was an important guide that we should do this. And when we sort of coalesced these ideas together, we wrote a short little white paper, I think that's got sent to some DOE people and sent to various people at the Joint Genome Institute. And pretty soon after we put together this idea, we immediately realized that we weren't going to try and tackle plants, animals, and fungi, both because they were so well sampled and because, certainly for plants and animals, the genomes were a little bit too big for us to propose sequencing thousands of them. So we excluded plants, animals, and fungi, and we proposed a genomic encyclopedia of microbes. Basically, I know that there are many multicellular things in various parts of the eukaryotic tree, but we lumped them all together and called them microbes, basically excluding plants, animals, and fungi. Sounds like protists, right? But we included bacteria and archaea in this too. And so we pitched this idea, and basically, very quickly, we were told, I'm sorry, eukaryotes are out. Their genomes are just too messy and too big. Bacteria and archaea have these nice little small genomes that you frequently are in one circle. We like your idea, but we're not even going to let you touch eukaryotic diversity. And we even said, well, but some of the genomes are small, and completely out. So we gave up on the eukaryotes eventually, although I must say, we had a great acronym. GEM, the Genomic Encyclopedia of Microbes. I even registered domain names associated with this, and then to be sliced out. And we argued forever about what the Genomic Encyclopedia of, we even said the Genomic Encyclopedia of not eukaryotes, and all sorts of other things in our discussion. I liked BAGEL, the Bacteria and Archaeal Genomic Encyclopedia of Life, or something like that. Believe it or not, it was nixed because someone said BAGEL had an ethnic, as in Jewish, ring to it. So we got stuck with GEBA, which is good because there's very little out there on the web with this acronym, so we kind of stand out. There's a Government Employees Benefits Association, which we've rapidly crushed with our Google search results. I think GEBA in Mandarin means something kind of weird, but that was beyond my ability to deal with. So we started what we called the Genomic Encyclopedia of Bacteria and Archaea pilot project, was what it was originally called, and it eventually became a full project. And the Department of Energy put their whole force of their microbial sequencing effort behind this. The Joint Genome Institute really took this on as one of their hallmark projects. And I sort of helped coordinate this from UC Davis, but really this was a DOE Joint Genome Institute effort. The people there that were behind this were incredible in terms of coordinating the project. The number of people involved was amazing, both Phil and Nikos, who did a lot of the overview, and Eddie Rubin and Jim Bristow, who gave support in a variety of ways behind it. I'll talk about this in a little bit. They threw what they call project management teams behind it, which were really incredible to have helping coordinate the project. Hans-Peter Klenk, after that initial conversation, as I'll tell you about, became our key partner in the project at the German Culture Collection, the DSMZ, and then Nikos Kirpides and Victor Markowitz coordinated annotation. There was the whole sequencing teams and closure teams that were involved, both from Walnut Creek and from the partners of the DOE JGI at Los Alamos National Lab and some at Livermore, and annotation was also done at least initially with people from Oak Ridge, and then a lot of people, including people in my lab and many people at DOE, got involved in analysis of these genomes. And there were even outreach projects, and Cheryl Kerfeld at JGI started an education project. I'll tell you about some of these. But it was incredible that we convinced them that phylogeny-driven approaches to sequencing genomes was a good thing. So what I'm going to tell you about for the rest of the time is sort of the project and some of the lessons we learned, and what I think we can apply many of these lessons to trying to fill in the eukaryotic tree. So what we did basically was we took the ribosomal RNA tree of life. That's where the best data is for phylogenetic sampling of bacteria and archaea. That's every cultured strain, or almost all of them, has a ribosomal RNA sequence available, small subunit ribosomal RNA, and of course there's all the environmental data from ribosomal RNA. So even though we realized that ribosomal RNA trees were not necessarily the best tree or the only tree, this is where the best data was. We threw out the eukaryotes, again, sadly, but we threw them out, and we took the ribosomal RNA tree of bacteria and archaea as our core piece of information. We went to GreenGenes database. This is where we both got the tree and got a lot of information about the tree. This is a ribosomal RNA sequence and tree database, but it has lots of other useful information in there, including information about whether or not something was a type strain or type species. And we really wanted to focus here on type strains, where there might be other information about that organism if we were going to sequence the genome. So then we linked up all the entries in GreenGenes with the Genomes Online database as much as we could, so we could find out what genomes were sampled already, how much of the tree had been sampled. And then we looked at the rest of the tree, and we asked, okay, are there branches in the rest of the tree that do not contain sequenced genomes? And then we asked, for those branches, is there a cultured representative available? We asked if there were cultured representatives in ATCC or DSMZ. DSMZ offered to grow the strains and isolate DNA at no cost. My budget for this project was $0. So they were going to throw sequencing at it, they were going to do annotation, they were going to throw a lot of people at this project, but we had no discretionary budget available to spend. And ATCC said they would charge us $6,000 to $8,000 per DNA sample. And we wanted to do hundreds. And DSMZ said, we'll do it for free. And not only that, they did DNA extractions, pulse field gels of the DNA, quantification of the DNA, separate cleanups of the DNA, and ribosomal RNA double-check sequencing of it. And this was an amazing partnership that we got via the DSMZ. So they became the main partner behind this project, largely because of their ability to collaborate at very low cost. So we merged all of this information together, and that's how we identified candidate lineages, candidate strains for sequencing. DSMZ, we identified about 400 of these originally. They targeted about 200 of these to grow and prep the DNA. Actually, maybe 300 of them. And then JGI, I made this slide a while ago, we're now up to about 300. But there's a key point in the process that's really important here. DSMZ, we sent them the list of things we would like them to grow. They grew the ones that they could. Some of them were either too hard for them to grow because they required weird conditions, or they didn't work. But that was okay. We realized that the important point here was phylogenetic coverage and speed and reduced cost. So if anything caused trouble, they just got thrown out. There was not a goal to sequence any specific organism. The goal was to have phylogenetic coverage in a large-scale project at as low cost as possible. And so we wanted to develop a pipeline that could be used in other projects. And this ability to just throw things out if they created any type of complication was really important in the long-term success of this project. If you care about an individual organism, you might spend half of your budget trying to get that organism growing, get good DNA from it, get good libraries from it. If it doesn't sequence well, if it doesn't assemble well, every stage of this process, if something didn't assemble well, they were gone. We did not spend enormous amounts of extra effort on any individual organism. So then the genomes got annotated, the data was released. This was an open community service project. So we convinced, or it didn't take much convincing of the JGI, but they agreed that the data would be deposited into GenBank as quickly as possible. And in fact, people published genomes on this data before we did. And we were completely fine with that. This was a community resource project to provide phylogenetic coverage for everybody. And then what we sort of decided was our task was to ask and answer a very simple question. Is providing phylogenetic coverage of genomes useful? I mean, I liked it. I want phylogenetic coverage of genomes, but we really wanted to show to people, or to ask, we didn't know the answer, whether or not phylogenetic coverage could be shown to have measurable benefits in various areas. So we wrote this one major paper on this project. There have been a bunch of other minor papers, but this one major paper came out in 2009 that basically tests this. And what I'm going to do is sort of show you some of these examples. Just going to skip over that. That's the phylogenetic coverage. We tried to cover diversity really well. We did. We claimed after the fact that we did this on purpose, that we were going to zoom in on a single phylum, the Actinobacteria, as both a test of breadth and depth. The truth is, Hans-Peter Klenk was the curator of Actinobacteria at the DSMZ. He knew how to grow many of these organisms. He had like 6 people working for him that all were the experts on these particular organisms, and he collaborated with everybody else outside of DSMZ who worked on these organisms. So when we sent him a list, you know, the Actinobacteria came back a lot more quickly. But it ended up being a really useful data set because we could test both breadth across phyla and depth within a single phylum. So again, the main thing that we tried to do was assess the benefits of this genomic encyclopedia, and that's what I'm going to talk about now. So the first question was very simple. We're marching our way through phylogenetic diversity using the ribosomal RNA tree. Does the ribosomal RNA tree actually identify novel lineages? If something is novel in the ribosomal RNA tree, is it novel, let's just say, in a genome tree? So that's what we basically asked. If we build a whole genome tree using a concatenated alignment of core phylogenetic marker genes and compared it to the ribosomal RNA tree, were the organisms that we selected still appear to be novel in the genome tree? It's a very simple question, but you know, ribosomal RNA does have its issues for certain taxa. You can have long branch attraction. You can have accelerated evolutionary rates in certain lineages. It was theoretically possible that ribosomal RNA was really, really misleading. And it turns out that was not the case. So we saw a pretty good correlation in terms of the PD, the branch length in the ribosomal RNA tree that identified something as novel, with the branch length in the whole genome tree. Wasn't a perfect correlation, but a pretty good correlation. So we were identifying organisms that at least their core housekeeping genes appeared to be novel, based upon finding novel ribosomal RNA sequences. Does that make sense? This is just a zoom in on the Actinobacteria portion, and we definitely, you know, every lineage that we sequenced appeared to be relatively distant from other lineages that either we sequenced or for which genomes were available. Now, this is not to say that the ribosomal RNA tree is perfect, right? The phylogeny, the phylogenetic patterns in the ribosomal RNA trees that we were using, I am sure contained some branching patterns that did not even reflect the evolution of the ribosomal RNA genes themselves, let alone the average evolution of the rest of the genome of those species. So, you know, there are many examples of this. This is one from when I was a tiger where Hyphomonas Neptunium in the 16S ribosomal RNA tree says it's a Rhodobacterales. In the 23S tree, or a concatenated alignment of phylogenetic marker genes, protein coding genes, it says it's in a different group, the Colobacterales. Now, it's still, you know, reasonably distant from other lineages in the 16S ribosomal RNA tree. That was our criteria, not its exact position. But again, the topology is not always perfect in these ribosomal RNA trees. So I think if we were to do this for eukaryotes, I mean, this is a question for many of the people here, what would we use to go through eukaryotic diversity and identify lineages that are phylogenetically novel? I would assume that the best data, the most data that is out there is still from ribosomal RNA for most lineages. There are clearly been surveys, PCR or EST surveys from across a lot of other eukaryotic diversity with other marker genes. And maybe for certain lineages like fungi or whatever, you might say move to other types of marker genes. But I think we probably could go through the eukaryotic tree and identify lineages that appeared to be phylogenetically novel compared to lineages for which genomes are available. I'd be happy to talk to people about how to do that, but I think we need to do this as a community. And maybe use other data for phylogeny. I don't think we should avoid using morphological data or physiological data or anything else that might tell us something about the phylogeny of organisms to identify ones that are, at least at a candidate level, phylogenetically novel. We're focusing on phylogeny, or I'm focusing on phylogeny here because, as we found out for the bacteria and Archaea project, everything else people were focusing on, physiology, location that they come from, pathogenicity, a variety of other features, and phylogeny was the one vector in diversity that was being ignored. And I think that's a similar case to eukaryotes. Phylogeny is the one main vector that is not being used as a main criteria still. So Nikos Kirpidis' group in particular tested a bunch of different ways the following question. So when we argued for the need for this phylogenetic sampling, one of the main hypotheses that we presented was that improving phylogenetic sampling of genomes should improve our ability to annotate other genomes. This is the argument behind sequencing other vertebrates as comparison to the human genome. That is, you have different levels of distance from your reference genome. That might help you identify rapidly evolving genes, medium evolving genes, slowly evolving genes, other types of diversity. And we asked a variety of questions related to this. Does phylogenetic sampling help us with a variety of annotation questions related to bacterial and archaeal genomes largely? And every way we looked at this, it seemed to be helpful. And we basically did a comparison of 56 phylogenetically sampled genomes versus 56 randomly sampled genomes. And every way we looked at this, there seemed to be a decent benefit towards annotation in general by having a better phylogenetic representation of genomes. Again, this is not to say that sequencing organisms based upon their ecology or physiology is also not important, but if you sort of control for that, phylogeny is a useful thing to have. I think this would probably apply to eukaryotic diversity as well. There's a separate issue, which is annotating eukaryotic genomes just in terms of gene finding can be a bit more complicated. So the benefit that you might get from having phylogenetic sampling might be ameliorated a little bit by having trouble actually finding genes in the first place in the different genomes to then use those to help compare to other genomes. But I still think phylogenetic sampling in general should help with annotation of eukaryotic genomes broadly. Another thing we learned, which we didn't really think about in advance but rapidly got interested in, was metadata is really important. So as we sequence more and more genomes, people would really like to compare thousands and thousands of genomes to each other or to some data set and ask questions about the organisms behind those genomes and whether or not they have features that you measure in the genome that correlate with the biology of those organisms. Well, who's going to go through all of the literature each time they do this and try and identify which organisms are virulent or grow on sulfur or whatever? So there's been a large effort by this group, the Standards in Genomic Sciences, to coordinate the collection, recording, and sharing of metadata about organisms. And the JGI has gotten really strongly behind that with recording information about the organism and trying to make it available in a database type format for people to use to do these comparative studies. As I sort of hinted at before, project management is absolutely critical for any of these large-scale projects, as I assume most people are realizing now, with sequencing being relatively easy and cheap. tracking samples and managing projects, both upstream of sequencing and downstream of sequencing, is becoming a very, very large headache, a very tough project. You know, if you're going to sequence 100,000 pathogen genomes, for example, as there's a project partly at UC Davis to do that, I mean, it makes me cry thinking about how to track all of the samples, where they came from, the data, the status of the projects. And there's no doubt that even for these 300 genomes, this project would have died without the JGI project management teams. They were the ones communicating with the DSMZ about where's the DNA, what's the quality of the DNA, we got the DNA, it's good, you don't have to send any more. We made libraries, we don't need to make new libraries. The annotation, the sequencing worked okay, the sequencing didn't work okay, and so on. And they would, you know, send me periodic emails about this, but they managed the flow of DNA, of organisms, of data, and so on throughout this project. And if we were to do something like this for eukaryotic diversity, there would need to be a significant effort in terms of coordination. You could still do stuff in a distributed manner, but someone has to track stuff and figure out what's sort of going on. Culture collections are absolutely essential for many reasons. They're sort of like the museums of microbiology, right? They know how to keep track of samples, they know how to store things, they know how to record vouchers and metadata. They also know how to grow many organisms that might be generally hard to grow. And also important, they know how to share the organisms after the genome gets sequenced. So the DSMZ, one of the reasons they participate in this project is now they sell the organisms for which the genome sequence is available. They also sell actually extra DNA if people want to do PCR or other things. But they, rather than us having to make the organisms available, they're making the organisms available, and it's, you know, now a community thing as opposed to an individual researcher's project. I didn't mention this in terms of the standards in genomic sciences, but another effort that sort of was linked to this project in a large way was what are called data publications. So you sequence a genome, you want to release the data to the community. It's very useful to have a description of how you generated that data, a description of the organism, how you assembled the genome, and you would like to get credit for what you did. So there's been a big effort to have what are called data publications, where you publish sort of a simple description of the project and the data. It gets a DOI and a citable unit, and then if people use the data, instead of just referencing a GenBank entry, which people rarely did, there's now a paper associated with that genome. And it just makes things just a lot cleaner in terms of using the data. So another really important scientific aspect for phylogeny-driven sequencing was we hypothesized and then tested that largely that we thought that this idea of lateral transfer wiping out all signal of diversity of organisms was not true, although we didn't explicitly test that. We tested sort of a counterpart to that, which was, in essence, what is the probability that you would find something new in an organism's genome? And is that probability related to how distantly related the organism is from other organisms that you have genomes from? So we basically asked, given, in essence, the PD, the branch length in the tree, compared to other organisms that you had data for, what is the probability that you're going to find something new in a genome entry? So we tested this in a few ways, and I'm just going to show you one of those ways. So we did what you could call a protein family rarefaction curve. We took data sets from multiple complete genomes, and we actually took the entire data set. We compared the data set, each protein to each other protein, and did a big clustering of all the proteins in all the genomes using this Markov chain clustering algorithm, this MCL algorithm. So we got a data set of protein families from across all the genomes that we were analyzing. And then we went and analyzed subsets of those genomes and asked, when we take subset number 1 and take genome number 1 and then genome number 2, how many new protein families show up in genome number 2 compared to genome number 1? How many new protein families show up in genome number 3 compared to genome 2 and 1? It's basically a simple rarefaction curve. And we did this for different genome data sets. So the first data set we did this for was Streptococcus agalactiae, which is not particularly important as for any purpose other than that this is the organism that was analyzed, leading people to invent the term the pangenome, and then do a lot of tests about what's called the pangenome. And the pangenome basically is, within a species, as you sequence more and more genomes, you see more and more genes that are not similar to any of the genes in the other genomes. And you know, this is very interesting in terms of biological diversity. And at the time, there were about 10 genomes of Streptococcus agalactiae, and each one that was sequenced was showing new genes. We did this slightly differently. We looked at protein families per genome instead of new genes. But you still see, as you add new genomes in this data set, new protein families showing up in each new genome within this species. So there is some genetic diversity at the protein family level for each new genome inside this species. However, if you do this for organisms that are more distantly related from each other, if you do this within a family, the Enterobacteriaceae, the rate of recovery of protein families per genome is higher than you see within a species. And if you do this within a phylum, the rate of recovery of protein families per genome is even higher. And if you do this in the genomes from our project, the rate of recovery is yet higher. The probability of finding a new protein family is higher if the organisms are further apart on the ribosomal RNA tree of life. Now, it turns out this doesn't really say an enormous amount about the importance of lateral gene transfer, but it does say something, I think, relatively simple, which is that synapomorphies exist for bacteria and archaea. That is, basically, that there are shared derived traits within branches in the ribosomal RNA tree. And if you sample things that are closely related to each other, you're likely to see a lot of these shared derived traits. And as you get further apart, you're less likely to see those shared derived traits, and you're likely to have organisms on very distant branches each have their own derived traits within their genome. We don't actually know if those came in by lateral gene transfer or by de novo invention of genes. But there is something about the backbone ribosomal RNA or genome tree that suggests that you will find novelty the more distantly related organisms are. So is that going to be true for eukaryotic diversity? I don't know. The indications seem to be yes, but we would have to test this. It may be true for some groups and not for others. It may depend on the extent of lateral gene transfer within certain lineages, just how true this is. But I think in general, the probability of finding new interesting things, if you control for sort of all other variables, or many other variables, you're going to find some novelty that relates to phylogeny and synapomorphies, shared derived traits within eukaryotic lineages that would make doing this worthwhile from a discovery point of view, from a biotechnology point of view, from for a variety of other reasons. So the third sort of scientific test that we asked was, we theorized that having phylogenetically diverse genomes should help us with analyzing metagenomic data. These are our references, our anchors for particular taxonomic groups. And if you scan through metagenomic data and you want to, for example, classify the genes that you find in metagenomes according to what their phylogeny is, the way we're going to do that is to compare to genomes. That's where almost all the data comes from different bacterial and archaeal groups. So if we don't have reference genomes for particular lineages, we're going to miss or misclassify things from metagenomes. So we theorized that by having these reference genomes, we should be better able to annotate metagenomes and better able to classify metagenomic data into what taxonomic groups came from. And I don't have the data from this, but we did basically a lot of analyses similar to things that Holly was talking about, also some just sort of annotation analyses to look at metagenomic data sets and then compare with and without the Genomic Encyclopedia genomes. What did we see? So we saw that these genomes, on average, helped us with a variety of metagenomic analyses. So will this be true for eukaryotes? By having, once we move more into eukaryotic metagenomic analyses and eukaryotic diversity, will having reference genomes help? Almost certainly. I'll come back to that in a minute. So as a spin-off from this project, we think we convinced largely the community that for bacteria and archaea, phylogenetic sampling is a decent thing to have. There have been multiple sort of, you could call zoom-in projects. We did one in my group, and the JGI did another one on the Haloarchaea. Ours was not directly related to the Genome Encyclopedia project, but the JGI one was a spin-off project. Cheryl Kerfeld at JGI ran a cyanobacterial zoom-in project. The paper was just published on this last year on filling in the phylogeny, going through the phylogeny of cyanobacteria and filling in genomes across the clade and what the benefit of that is. We've been doing this again for Haloarchaea, as has JGI, and I'd be happy to talk to people about that too. There's a sort of phylogenetic survey, although it's also a functional survey of root nodule bacteria at the JGI. So I think there's a lot of these surveys, and we convinced a lot of people that this is good to do for bacteria and archaea. However, I'm going to go back to this for a minute. The GEBA project definitely seemed to improve metagenomic analysis, but barely. I mean, like, we could detect it. Many of the tests were not statistically significant, and a few of them were. But it was borderline trivial improvements in metagenomic analyses. And I'm going to tell you why, I think. So I think the first reason why is that the methods that we were using to analyze genomic and metagenomic data were not really attuned to having lots of genomes or having phylogenetically diverse genomes. So we need to make better use of the data. We need, I mean, I could discuss all sorts of things that we need, but we need improvement in the methods, improvement in the data sets, more simulations to figure out which methods are appropriate to use given that you have phylogenetic sampling. And we've done a lot of this in my lab or in collaborations in which my lab was involved. We had this large ICEAM project, which was funded by the Moore Foundation, a collaboration between my lab and Katie Pollard's lab and Jessica Green's lab. And we tried to develop a lot of computational methods that would improve our ability to analyze metagenomic data. And part of this was improve our ability given that we would now have phylogenetic sampling. Steve Kembel developed a correction for ribosome RNA copy number. Tom Sharpton developed a phylogenetic approach to identifying OTUs. You've already heard about PhyloSift. Steve Kembel developed a way to classify sequences using all the reference gene markers at once rather than just one at a time. We've been trying to build protein families from metagenomes. Our latest stuff has been to work on identifying more markers, in particular markers for individual phyla rather than for across all bacteria and archaea. We need a better reference tree, and this is no longer submitted and it's no longer Morgan, but Jenna Lang has a paper that just came out in PLOS One on building better reference trees. We've built a data set of protein families from across all genomes that we call SFAMs that's also been published. We've developed methods that are trying to automate the process of masking. And we're not the only people. There are lots of people here and lots of people in the community that are now, I think, tuning their methods to make better use of full phylogenetic diversity of reference data. I just was showing some examples of the ones we've done, but there's lots of other stuff out there. But the last thing I really want to leave you with is the real reason I think that this completely failed, in essence, to help with metagenomic analysis. And that's that we've barely touched diversity in terms of this project. So if we go to the ribosomal RNA tree of life, for example, we can measure the PD, the phylogenetic diversity in different groups. That's the branch length in the ribosomal RNA tree. And in our paper, we showed this. So if you take all the previously sequenced genomes of bacteria before our project, and you ask, how distant are they from each other on the ribosomal RNA tree? How much of the ribosomal RNA tree do they cover in terms of branch length? And then you sort by the max PD, and you get a plot like this, where many of the genomes, as you add more and more genomes initially, they're pretty distant from each other as you go through the tree. And eventually, you start to saturate. We have 19 E. coli genomes and 23 anthrax genomes, and they don't add much PD when you add more and more of these genomes. So when we did our project, each genome that we added a lot of PD to the total data set. It better have, that's how we selected them. So if this didn't work, our initial algorithm was totally screwed. If you look at the Berge's manual, basically, the cultured organisms in the bacterial clade, and you ask how much PD do they represent in the ribosomal RNA tree, it's quite a bit, but you could capture about half of this PD, half of the total phylogenetic diversity of cultured bacteria and archaea with only like 2,000 or 3,000 genomes. And again, with sequencing now, that's completely doable to capture half of that diversity. In fact, largely because of this plot and because of acceleration of sequencing, they are going to sequence everything. Every type strain is now being done as part of a project at the JGI. However, if you look at the uncultured diversity, the ribosomal RNA sequence is not associated with cultured organisms. And at the time we did this, this was just full-length 16S sequences in the GreenGenes database. This data set has probably expanded 100-fold since we did this in terms of uncultured organisms, and not at all in terms of cultured organisms. To capture half of the uncultured diversity, again, in this incredibly small sample of total uncultured diversity, we would need 10,000 genomes. And that's 10,000 genomes of uncultured organisms, which we didn't have any idea how to generate at the time. However, methods have gotten better, and now there are many different ways where you could imagine generating either reasonable assemblies or even complete assemblies of genomes of organisms that have never been cultured. Of course, you can get them into culture or enrichment cultures, but you could do shotgun metagenomics from low diversity ecosystems, or maybe, if Titus tells us how, from high diversity ecosystems, and build assemblies from any organisms that way. There's been a lot of effort in doing sort of single cell sorting or amplification to generate genome sequences. There's even a large GEBA Uncultured project now at the JGI. Tanya Wojcicki has been running. The first paper on this project has now been accepted. But there's a large project that they are coordinating there to march their way through the real tree and fill in genomes using single-cell genomic approaches. Can we do the same thing with uncultured eukaryotes? I think certainly. I think that many people here know a lot about the diversity of uncultured eukaryotes, and we could start to think about how we would obtain partial or full-length genomes from a lot of them, and I know some people here actually work on that. The last thing I want to say is, of course, just sequencing genomes is not enough. We need experiments from many of these lineages, either the organisms themselves or the genes that come from those organisms. Because, in fact, although the genomes were biased when we started our Tree of Life project, the experimental studies were even more biased and still are. Most of the experiments are still from 3 phyla of bacteria. There are a few other phyla that have a reasonable number of experiments, but it's still incredibly biased sampling of experimental studies. And the same is true for eukaryotes and viruses and archaea. So we need experimental studies from across the tree. I don't know if people have heard of this Adopt a Microbe campaign that Cheryl Kerfeld was running at the JGI. For the Genome Encyclopedia genomes, she tried to coordinate high school and college courses that would adopt one of the organisms to then either do bioinformatic analysis of the organism or actual experimental studies of that organism. And they get to play with a genome that's distantly related from anything on the planet, has been barely studied, and they've done some really cool things with that project. So what I think at this meeting, I think it would be good to think about is, if people agree that this is a useful thing to do, is how to do it. We tried when I was at Tiger to organize a protist genome sequencing consortium. We even put in a few grant proposals related to a big coordinated effort to do protist genome sequencing. They were all rejected. Many spin-off projects came from that, so I got involved in the sequencing of the tetrahymena genome. Many other protist genome projects came from that original meeting that we had at Tiger in 2004 or something to that effect. But I think the time is right, given sequencing now, to get the community together to organize an effort to really march our way through eukaryotic diversity and sequence the genomes, provide them to everybody in an unrestricted manner so that we're not all competing with, fighting with each other about when am I going to see the data, and make this a large collaborative community effort. And I think we can do that. So I will end with acknowledgments. As I've said before, the GEBA project was this amazing collaboration, including all these people at JGI, many people in my group, Jana Lang, who's here, Dunying Wu, who's not, Aaron Darling, who's not, and a few other people helped in a lot of ways with that project. The GEBA Cyanobacteria project, this paper just came out last year, and you can see that largely Cheryl Kerfeld's effort. The Halo Archaea stuff that we're doing in my lab is a collaboration with Mark Facciotti's lab here at Davis. This iSeam project was a great collaboration with Katie Pollard and Jessica Green, and led to lots of cool sort of metagenomic informatics. This Tree of Life project, which kind of died when Tiger imploded, was still a fun project while it lasted, and Naomi Ward was really the main player behind it. And I'd like to thank lots of other funding agencies and everybody else in my lab and you guys for listening.